Next speaker, it's Morgane Thomas-Chollier from IBENS. Morgane, can you?
Yes.
Put your present. Yes, it's okay.
Is it? No, I guess it's not full screen. I'm putting it full screen.
Yes.
Is it good now?
Yeah.
Okay. So good afternoon, everyone. First of all, I'd like to thank the organizer of the symposium. I'm really happy to be here today to talk to you about motif discovery and about our tool called RSAT. So I wanted to explain a bit about the Neuromotive discovery, just to be sure that we are all understand the same thing. So here, I take the example where we have a few target genes that are co-expressed genes and that are co-regulated actually because they are bound by a transcriptional factor, here represented by the blue circle, on cis-regulatory elements, so the transcription factor binding sites, here represented in orange. And out of that, what we want is to actually find the binding motif that is common. So if we rephrase that, the idea is, are there some overrepresented motif in a sequence data set? So to go more into the details, the motif discovery problem actually is when you have a set of sequences like presented here. So here we have upstream sequences of the few genes that I showed you earlier. And we would like to know if there is a common regulating transcription factor. Can we discover its motif on the basis of just the sequences? So just to be clear, we have the sequences. We suspect that they share some functional signal, but we're not sure. We don't know the transcription factors that are involved, and we don't know the cis-regulatory elements. So the motif discovery problem is a really tough one. And it's not to be mixed up with what Francois just presented, which is actually, which falls under the pattern matching problem. So the pattern matching problem, you have a genomic sequence here. You have already a motif of interest in form like Francois presented of a PSSM or a TFFM or a Kmers. And the question that you want to address is there a putative cis-regulatory element in the genomic sequence? And if so, at which sequence? At which position, sorry, in the sequence? So in this case, we do know the motif of the transcription factor, and we want to search for matches of the motif in the sequence. So this problem is known as the motif scanning problem. So today, this is not about that we're talking about, but about the motif discovery problem. So actually, this problem is really not recent. One of the main tools, probably one of the most well-known tools to perform the Neuromotive discovery is MIM. And MIM was developed, as I showed here, in 1994, so more than 20 years ago. And it was meant to discover motifs in proteins and in DNA. And the table that I took here is actually from the paper. And they show here that what they've been doing is testing the program. At the time, there were not so many genomes that were available. So they were working on the bacteria, Escherichia coli, and they were interested in the binding sites of the CRP transcription factor. And what you can see here is that they were testing it on 18 sequences, and the average length was 105 base pair. So really, really small data sets. And a few years later, another algorithm came out this time. It was to work on the yeast genomes, the Saccharomyces cerevisiae, and the binding sites. And again, here I took information on the data set on which it had been tested at that time. And what you see here, it was actually on 7 genes that were co-expressed in response of particular nitrogen sources in the medium. One advantage of this approach was actually that it was a Kmer-based overrepresentation that was computationally more efficient than the program MIM. So this program was called Oligoanalysis, and it soon became the core of the suite that is called RSAT. So what is RSAT? So RSAT means Regulatory Sequence Analysis Tools, and I think we can fairly say now that it's a classic vintage, mature, not to say old, suite, because it's started in 1998 and we celebrated the 20th anniversary two years ago. And this suite was initiated by Jacques Van Elden, so the same Jacques that spoke this morning and that is co-heading the Institut Français de l'Informatique. And the two main tools initially were Oligoanalysis that I just presented, and another Neuromotive discovery program that is called dyad analysis, and that's actually specifically made for space motif. So along the years, many contributors went to work on RSAT, some pictures here, not all of them. And currently the suite is jointly headed by Jacques, Alejandra Medina Rivera, who is in Mexico, and myself. And today I'm going to show you mostly the work of Walter Santana Garcia, who is in the lab. So there has been many improvements on RSAT along the years. Here are the main publications that we did. And I like to show the evolution of the website from these really pinkish colors in the 80s towards what is currently our website. So importantly, even if it's, let's say, classic suite, actually it has been extended with many developments that were directly prompted by advances of the field of regulatory genomics. So here what you see is a timeline that we published in our anniversary paper in NAR 2 years ago. And one really important milestone is the advent of ChIP-seq. So François already explained what is ChIP-seq earlier, and indeed this has really been a major change in the regulatory genomic field. So what does it mean for the de novo multi-discovery problem? Because indeed it totally applies to ChIP-seq fragments. So if I take the same illustration as before, the difference is that instead of having the genes as reference, this time we have fragments of DNA, and they constitute the peak data set on which we want to perform de novo multi-discovery and discover the binding motif. So it means that at that time, clearly new approaches were needed, specifically for the ChIP-seq data set. And the main reason was the size, and the size of the data set to analyze. So I show you again here the little table from the mean paper. And just to recall, it was tested on 18 sequences of an average of 105 base pair. When we had the first ChIP-seq results, we actually realized that it was dozens of thousands of peaks and dozens of megabase per sample. So it was clear that the programs, at least some of them, were really not able to scale to this new avalanche of data. The second problem was that, well, the actual multi-discovery problem criteria was somewhat different, because we used to work with promoters, so proxy as upstream sequence of co-expressed genes between 200, let's say, and 2 kb. And now we had peaks that were a few hundred base pair, but with importantly positional bias, which means here on this illustration, that if you take the peaks and you align them on the summit, that the binding motif of the transcription factor is more likely to be near the summit than on the flank. So that means that it's an information that we can use to actually discover the motif and not just the sequence themselves. And the last point was that we could see that lots and lots of labs were moving towards ChIP-seq, and that motif analysis was really meant to be not for specialists anymore, and that we needed user-friendly workflows that were much more complete than just finding the motif. So at that time, so we developed in RSAT a program called peak-motifs that was dedicated to motif discovery in ChIP-seq datasets. So how does it work? So we start from peak sequences, And as you can see here, it's written complete data sets. This was important. I will come back to that in a few minutes. And the program applies multiple de novo motif discovery algorithms. So it runs oligoanalysis for global overrepresentation. It runs dyad analysis for the space motif. And we have two other programs that takes as criteria dispositional bias that I told you about. So the program finds this motif and then continues the workflow so that the user actually gets a lot of information. So first it locates the motif. So this time it's really this motif scanning problem. So it locates the motif on the peaks, and it's possible to visualize the binding sites like here in green on tracks in genome browsers. So here it's UCSC, but you can also do it in Ensemble or FGV. And very importantly, it also automatically compares the discovered motif to the database. So we have 50 collections in RSAT of public database to try to annotate those discovered motifs. You can also provide your own motif database, for example, if you have a license for transfer. And then what you can do is visualize the discovered motif. So for example, here with logo alignments with the known motif from databases. So what was peak-motif really important at the time was that it was able to treat full-size data sets. Probably today it's a bit weird to show it like that, but at that time it was really an issue. Most of the ChIP-seq data sets were analyzed only with the top 200, top 300 peaks instead of the 30,000 ones, simply because the motif discovery programs were not able to treat that many. So I like to show this plot because it illustrates really this conclusion. What you see on the x-axis is the sequence size. And this arrow shows a typical ChIP-seq data set of around 25 megabytes. And the size limit of the websites most of the time was around 5 megabytes. And one of the reasons is that if you look now on the y-axis is the time, the computing time in seconds. And this red horizontal line here corresponds to 1 hour. And I've told you about mean, and mean actually is this line here, in the gray dotted line that is on the y-axis, because it really is not able to scale at all, because the algorithm was not made to be like that. So it's really hard to use the original mean algorithm on ChIP-seq data. And it was actually the same as the Chipman problem at that time. And interestingly, the Kramer approach that was oligoanalysis and the other programs in RSAT were actually scaling really well with the large data. So that's why we had this advantage that it was actually fast enough that we could propose to treat the full-size data sets from the website. And that was really, at that time, very important. And we were providing a complete pipeline. Importantly, because we were able to treat the full-size data set, we were able on test data of the OC4 and SOX2 ChIP-seq data sets that were also really often used for benchmarking. We were able to find alternative motifs that were known in the literature, but that were not known or not, let's say, discovered by the other programs because they were only looking at the top peaks. So since then, because this is 10 years ago almost, many new programs have been developed. So NIM has done a new ChIP approach, which actually embeds a program really similar, almost the same as oligoanalysis. Chipman, it's Motif, Omer, and I'm not going to leave you all of them because really many were developed. And interestingly, the machine learning approach were also applied to this problem with starting with DeepBind in 2015, more recently TFU Disco. Again, I'm not going to list them all, but what is clear is that performance is really high, it's really good. But the adoption actually of this tool is not really wide. So one of the reasons is that it's difficult to interpret the models. And also a real burden is the computing efficiency. So it's clear for us that non-expert users that are actually producing their data sets, they need to be able to autonomously analyze their data sets without having a really big computing cluster next door. So we are convinced that there is still a real need for reliable multi-discovery tools that are accessible through a web interface dedicated to non-expert users. So this is exactly what RSAT is. So it's accessible to non-expert user website already from its origin. And it supports more than 10,000 organisms. And we have freely accessible servers organized for biologist communities. And we have documentation, tutorials, and help. So now if we focus on Motif itself, after 10 years of existence, what we can say is that there are really some limitations. The first one, that's probably the most obvious, is that the web interface and the result reports are clearly out of date, and they could be presented in a much more accessible way for the user. Repetitive regions like simple repeats in eukaryote genomes can lead to erroneous incorporation of these sites. So this is a problem that we noticed. Another one is that because we use this bias, position bias as criteria, sometimes we do find underrepresented motifs that are reported but mixed with the overrepresented motifs. So for the user, it's not always really easy to pinpoint this. And there are new features that we wanted to add, like clustering of the resulting motif. We want to improve the ranking system. And many users have asked us if they could give as input a bed, the bed file, for example, that comes out of Max. Currently, we only accept the FASTA sequences. So that's why we decided to go on and develop a peak-motifs 2 version. The first thing was to update the form. So this is more for layout. So this is the previous layout that is 10 years old. And now we have moved to this new layout that was already used for other RSAT tools using Bootstrap. So everything that I'm going to show now is the work of Walter Santana Garcia. And in the form, we have added, as the users have asked, this option to upload bed files and then retrieve genomic sequences from the genomes directly. So you can choose, for example, a UCSC organism, and you have within the interface the choice of the genome that you can choose, or you can use a RSAT installed organism. And we have added this masked low complexity region to be able to remove those repeats that are entering the results. The report is what we've been working on a lot. This is the previous layout, really outdated, as we were saying. So now we move to a new layout that is more like a dynamic dashboard. This is what the users are more used to with other tools. And so what you see here are different panels. So the settings, the quality control, discovered motif. And these are screenshots from the settings. So now it's much easier to, so for example, we have been informed what input parameters have been used to recall the files that have been put as input. And we have this quality check panel that was the first panel in the previous layout, but that was often overlooked by the users. So now we have updated it, and I think it's clearer for the users that that is important. And the discovered motif, this is the previous way where we were showing each motif, lots of information. And now we have moved to a table that is dynamically ranked. So we are making progress in that. So, and yeah, sorry. And when you click, then you can have like more advanced information on each of the motifs. You can directly download the publication quality logos or matrices in a standard format. You can also send it to Convert Matrix, which is another program in RSAT where you can even have more formats. So what we are still working on, this is the blue buttons here. So this is not finished, it's in active progress. So it's the comparison of motifs. So this was already implemented in the previous version, and which we need to put that again. And two other points, which is the clustering and the enrichment of motifs. So for the clustering, as I was saying, we output many motifs, and some of them are really similar. So we need to create non-redundant list of motifs out of peak-motifs. And for that, we already within RSAT have a matrix clustering program, which is called matrix clustering, developed by Jaime Castro-Montagon. And this program works really fine. So here you have how it works. And we showed that it's really usable to facilitate the interpretation of results when you have like lots of motifs, and you can easily collapse them and cluster them. So I will move on that. The idea here is that the program is working really fine, but we need still to plug it inside Motif so that it's completely seamless for the users. And the last point that we are working on is trying to have a new ranking system here, because the current ranking system is on the significance values of the k-mer, and we know that this is biologically not always really the most relevant. So we've been working on another program that is still in early development that I wanted to show you so that you have an idea of where we are going in RSAT. So the tool will be called Matrix Centrality. And the idea is to use motif scanning and specifically focus this time on what's happening at the summit. So what you see here is a quite simple plot where you have the distance of all the predicted sites toward the summit that is in the middle. And the idea is to actually calculate the difference between the highest point and the mode of the number of predicted sites. This panel B is actually what we call the unibind plot. And I'm sure Anthony will talk to you about that, because this is a plot that is actually made in unibind, that takes into account the p-value of each motif and the distance to the summit. And in this example here, you can clearly see that there is a clear enrichment in around the summit, let's say, of the peaks. And you also see here this violet density that shows that it's enriched also in the center. So then what we do is automatically create heat maps where you have here all the motifs output by peak-motif, and you can actually rank them depending on the enrichment at the center. You can clearly see here, so in red is when there is an enrichment, and in blue it's when it's depleted. So this also fixed the issue that we had where we have some underrepresented motifs that the users had issues keeping them. So in a nutshell, RSAT does a lot of things. I've talked to you today about the Neuromotive discovery, but we have many tools to other things with Motif. And I will end with this slide. One of the current progress that is more technical that we have been doing is to actually move the code to RSAT to GitHub as well as our documentation. Jacques has been working a lot on Conda installation. It's not totally finished, but we are pointing to the end of this program. And we have REST web services. So if you want to know more about the Motif tool, you can go and see the poster 26, which is the poster of Walter. So thank you very much, and I'd be happy to take your questions.
Thank you, Morgane, for this speech about peak-motif 2. Just one question before other question. When you make a clustering of motif, did you find a core motif that correspond to each other motif? Did you study that, or just?
So I went a bit fast on that, but this is exactly what you can see here on this example. For example, here in the red cluster, you can actually see by eye that they really go together, and you can actually collapse them into a single one that is here represented with the same red square. Interestingly, when you do that with all these really big clusters, so this is the OCT4 SOX2, quite common example, you can really see the variations of the two. So we tend to be more interested in what is actually different than what is completely common, because when it's common, we try to collapse and remove it. But I guess we can see the other way around.
So perhaps you could associate to your cluster matrix a very small motif that which each position is conserved or not? You don't know?
Yeah, I mean, you could do whatever you want. On this example, actually, we are mixing motif obtained by mean, motif obtained from RSAT, and I think even motif from Omer. And we wanted to be able to integrate all them and see how much they were similar. Okay. So we have this idea of collections on which that you can tag. So you can put a collection of an updated motif or whatever you want. Yeah.
Okay. Other question? We have time? Yeah, François, go.
So thank you very much for the talk and for the effort in developing those tools, because they are super useful for us. And my question is about the underrepresented motif. Do you think they have any biological significance, or they are just consequences of overrepresentation of other ones? Like if you have many GC in the real one, you will lack the 81, for example. So do you think there's any value of this underrepresented?
Yeah, actually, for a while, we were really skeptical, and I had the chance to work with a collaboration with Sebastian Meissel when I was in Berlin at the Max Planck Institute for Molecular Genetics. And we actually dissected biologically one of these underrepresented motifs in the context of the glucocorticoid receptor binding. And actually, we managed to show that if we were taking these motifs that were evicted and we were putting them in a reporter assay, it was preventing the binding of the glucocorticoid receptor. So one of the hypotheses of Sebastian, who is a biologist, wet lab biologist, was that it was linked to the paraspeculums thing in the nucleus. I have to say I'm not a specialist of the paraspeculum, but he had this impression that this could be linked. So I think it's something that has not been studied a lot. There seems to be not so much specific, I mean, the motif didn't have to be so specific to remove the binding of the GR, but clearly there is a biological way. And I did extensive bioinformatics analysis in this paper to show that it's not just a problem of composition.
Thank you very much.
Question? Yes.
Yeah, thank you, Morgane. Really nice talk. And yeah, you know how much I love RSAT. There is no more to say about that. I was just wondering whether you planned in the future potentially to incorporate methylation information. Because, I mean, François have shown a bit, I mean, he touched really briefly upon that, and there are many publications getting published mainly recently, where, you know, they have expanded the alphabet, where instead of ACGT, you have also unmethylated cytosine, methylated cytosines, also can depend on the context where they are CpG or not. And that could also be interesting to incorporate that in RSAT, and I think that should be quite easy, I hope, to just extend the alphabet, right, and try to learn basically what are the motifs, also considering methylation information.
Yeah, so there are 2 points to answer the question. So the first one is really technical. We have to update our tools to be able to take multidimensional matrices. So this is something that we've been talking for a while now, especially because we have another collaboration with Twati Craft in Canada. And Twati actually works on methylation and is really close to the RSAT team. And he's developing a database of motifs that have differences in terms of methylation. So he would like that we have these in RSAT. So we do have it, but not with the information of the methylation, because currently our tools are not made for that. So this is definitely something that we want to add. Yeah, very good question.
Okay, so we could